All right, today we're cracking open the case files on some of medicine's most notorious microscopic criminals. These are the culprits behind some really perplexing medical mysteries. So, let's dive right in.
Okay, so what do a sore throat, a cat bite, and a faulty heart valve have in common? I know, it sounds like the beginning of a very strange joke, doesn't it? But the answer is actually pretty fascinating.
They can all be crime scenes. And that's what we're going to do today. We're going to profile a rogues gallery of 5 unique bacterial suspects, and each one has a totally distinct method of attack, their own MO. First up, the factory saboteur.
So let's meet our first culprit. This one is an absolute master of industrial sabotage, but on a cellular level. It doesn't just make you sick, no, it shuts down your body's most essential factories right from the inside out.
Okay, so here's the rap sheet. The suspect is Corynebacterium diphtheria. It's wanted for causing diphtheria. Its weapon of choice, a seriously potent poison called the diphtheria toxin. And its signature crime is bringing the protein factories inside our cells to a complete, grinding halt.
So how does it pull this off? Well, the attack is incredibly specific. The toxin uses a process called ADP ribosylation. Think of it like jamming a very specific, very important lock with the wrong key. It targets just one vital piece of cellular machinery, this thing called elongation factor 2, or EF2, and completely shuts it down. And with no EF2, you get no new proteins. And if a cell can't make proteins, it dies. It's like a perfectly targeted assassination at the microscopic level.
And this is exactly what makes it our prime suspect for this particular crime. See, other toxins, like the ones from cholera or pertussis, they hijack cell signaling. The Shiga toxin attacks the ribosome itself, but only the diphtheria toxin carries out this super precise sabotage of EF2. Case closed on that one. Now, let's move to our next case.
All right, next up on our most wanted list, we've got a real opportunist. This is a culprit that takes something super common, like a sore throat, and just escalates it into a life-threatening emergency.
Meet Fusobacterium necrophorum, the #1 bad guy behind Lemire syndrome. The crime always starts at the scene of a simple throat infection, but its MO is way more sinister. It causes a dangerous, infected blood clot in the internal jugular vein.
And here's where it gets really dangerous. It's all about the chain reaction. The infection breaks out of the throat, invades the bloodstream, and then forms these nasty clots in a major vein in your neck. From there, little pieces of that infected clot can break off and just travel all over the body, causing absolutely devastating secondary infections, like abscesses in the lungs.
Our next case file starts with something that could happen in any of our homes, a bite from a pet. But lurking in that seemingly small wound is a hidden and very fast-moving danger.
The perpetrator here is Pasteurella multicida. It is the most common pathogen we find at the scene of a cat bite. And its signature crime? A soft tissue infection that develops with incredible, almost alarming speed. We're talking it can just explode within a few hours of the bite.
Now, it's super easy to get this suspect confused with a few others in the animal-borne lineup. But like any good investigation, the forensics are key. Bartonella henselae, that comes from a cat scratch, not a bite. Capnocytophaga, that's usually linked to dog bites, and Streptobacillus, that's from rats. So for a fast-moving infection from a cat bite, Pasteurella is our microbe.
Okay, now we're getting into the really tricky cases. These next two are the masters of disguise. We're talking about the ghosts of the microbial world, famous for evading all the standard detection methods.
So, the big challenge they throw at us is something called a culture-negative infection. What does that mean? Well, it means a crime has been committed, you can see the damage, but when you run the usual tests in the lab, nothing grows. It's like the culprit was never there. You're basically chasing a ghost. And to catch these guys, investigators have to pull out some much more advanced forensic tools.
So our first ghost is Trophorema whippley. This one's wanted for Whipple's disease, which is a really nasty multi-system illness that can cause everything from weight loss to serious joint pain. And since it won't show its face in a culture, detectives have to hunt for its telltale forensic clue, specifically stained cells that show up in a biopsy of the intestine. And our second ghost? Oh, this one is even more notorious. Meet Coxiella burnetii. It's a prime suspect in culture-negative endocarditis. That's a really dangerous infection of the heart lining, especially when it happens around prosthetic valves.
It's also well known for its connection to Q fever.
So how in the world do you catch a killer you can't even see? Well, the investigation usually starts when a patient has a fever and some heart valve problems, but crucially, the lab cultures come back totally empty. That's the huge red flag. So investigators bring out this special gear, serology to look for the antibody footprints the culprit leaves behind, or PCR to look for its actual DNA. And boom, that's how they unmask Coxiella.
And if you look at the lineup of suspects for this particular crime, you can see just how important it is to know who you're dealing with. Yeah, other bacteria can cause culture-negative endocarditis, but the data clearly shows that Coxiella burnetii is one of the most common perpetrators, especially when there's a prosthetic valve at the crime scene.
All right, let's do a quick debrief. Let's run through these case files one more time. We've got the factory saboteur, diphtheria, the throat infection opportunist, F. necrophorum, the rapid response attacker from a cat bite, P. multicida, and our two masters of disguise, T. Whipley and C. burnetii, who both require some pretty special detective work to unmask. 5 different culprits, 5 totally distinct MOs.
And really, that's the big takeaway here, isn't it? In medicine, just like in forensics, identifying the criminal depends entirely on understanding their unique methods. It is the absolute key to cracking the case and making sure the right justice, or in this case, the right treatment, is served. Which leaves us with one final thought. We've cracked these 5 cases, but the microbial underworld, it's always changing, always evolving. The next great medical mystery is already out there, just waiting for a sharp detective to figure out its MO. Thanks for joining us.